KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Education Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Good morning students. In this video, I would like to continue about our previous chapter. We are started digestive system of human beings. In that previous videos, glands of digestion, digestive glands. In the digestive glands, salivary glands we completed, and then gastric glands, intestinal glands we completed. Okay. In this completed glands, intestinal and gastric glands are located along with alimentary canal wall. Correct, na? And then salivary glands, pancreas and liver, these are the associated glands. Okay. So in this video, continuation of the previous video concept, liver is one of the gland, digestive gland, and then pancreatic. These glands onwards, what are the enzymes are secreting, that glands juices, what is their composition, what is their pH. Along with that, the conclusion I am started about physiology of digestion. Physiology of digestion. These two concepts we have to discuss in this video. Let me start today video concept. Dear students, see, and yesterday liver is located at the place of the below the abdominal, in the abdominal place below the diaphragm, that to right side. It contains the two lobes that we completed. So that introduction I will say one more time. Liver also called as a hepatic, the nickname of liver, scientific name. That liver having the two lobes, that two lobes, hepatic lobes, that hepatic lobes inside hepatic lobules are there. That hepatic lobules are inside, they are arranging several cells are in the sequence order. That sequence order cells we call the hepatic cords. That hepatic cords contain the hepatocytes. These hepatocytes are synthesis a fluid. That fluid we call as a bile. That fluid, bile, is playing important role in the emulsification of fat. Okay, then here that bile totally is drained into the in between the hepatic cords. Some cavity is there. That cavity we call as a sinusites. Okay, hepatic sinus. Okay, then now what is the significance of liver functions? Okay, liver functions. See, basically liver functions, liver contains, liver secrete bile. That bile contains two important substances. One is the bile pigments, another one is the bile salts. Bile pigments are names bilirubin, biliverdin. These are the example of bile pigments. Pigments means colored substances. And then bile salts, glucocholates and taurocholates. Sodium, potassium glucocholates and taurocholates. Bile pigments, what is the names? Bilirubin, biliverdin. These bilirubin and biliverdin are bile pigments are formed by the breakdown of hemoglobin. When the RBC cells lifespan is over, RBC lifespan 120 days kadana. In that 120 days completed after, that cells will come to the liver. In that liver, hemoglobin extracted, then that cells extracted cells, separate from the RBC cells onwards hemoglobins are extracted, then that cells are going to the spleens itself is destroyed. So that's why RBC cells are destroyed by the spleen, so that's why spleen we called as a graveyard of RBC. Okay, and then before reaching of the spleen, it will reach the RBC cells reach the liver onwards. In the liver hemoglobin extracted, that hemoglobin breakdown after it create the pigments. That pigments only act as a bile pigments, bilirubin and biliverdin. Now you got the point. Okay, because of hemoglobin is a respiratory pigment, colored substance, correct? No? Okay, then here these bile in the yellow in green color, yellow and green color. Okay, slightly yellow and green color. Now in this situation I will be recall one of the situation in our life. When we are getting the vomiting sensation, when we are showing the vomiting, on that time undigested food will come out from the stomach, and then that contains a lot of mucus. That mucus totally in the form of the yellow and green color because of it is presents the bile, and along with that the taste of that vomiting substances in the bitter in taste, bitter, correct na? Okay, so that is causing by the bile presence or undigested food in that vomiting undigested food. Okay, so here bilirubin, biliverdins are produced from the breakdown of the hemoglobin. Now another function, carbohydrates metabolism, very most important thing. Okay, liver play an important role in the carbohydrate metabolism. That carbohydrates after digestion it will produce the glucose, that we know. That glucose onwards it will produce the some derivatives. Okay, some derivatives. What are that? Here carbohydrates metabolisms wise liver it will play an important role in the glycogenesis. So glycogenesis means after digestion of the carbohydrates it produces the glucose converted into the glycogen, in the place of the liver. That process we call as a glucogenesis. Okay, glycogenesis. Okay, sorry, my pronunciation is glycogenesis. Recall glycogenesis. Genesis means production. What it is producing? Glycogen is producing. Which substance onwards? Glucose onwards, glycogen production. So what we are taking food, that food having the excessive glucose, that excessive glucose stored in the form of the glycogen in the place of the liver. Okay, reserve food. When we are not taking any food in the some span, I mean 1 day, 2 days, this glycogen, reserve food is there now, that glycogen again converted into the glucose. Okay, that glucose, that amara nirahara deksha, something in Tamil what it is I don't know. Continuously some of the politician people does not take any food. They are doing the something revolutionary activity by the some things. Okay, the thing on the time they does not eat any food, fasting, nothing but fasting. In the time of the fasting time, the reserved food glycogen is converting into the glucose. Okay, that process glucogenolysis. Glycogen onwards glucose formation we call as a glycogenolysis. That means glycogen lysis. Glycogen is breakdown into the form the glucose, that process. Okay, and then next one, gluconeogenesis. Gluconeogenesis means glucose will form in which substances onwards, from the which substances other than carbohydrates. Basically carbohydrates onwards they are producing the glucose. Okay, after digestion, that we know. But other than carbohydrates particles, nothing but amino acids and fat. So fat and amino acids is and all not at relation with carbohydrates. But when our body is required, when our body faces the low glucose content, on the time our protein, fats, these and all other than carbohydrates particles, that onwards the glucose production we called as a gluconeogenesis. Neo means following. Okay, just you have to remember like that. Gluconeogenesis, the glucose genesis by the other than glucose particles, carbohydrates particle. And then lipogenesis. Lipogenesis, what we are taking food, that taken food, any fat is there, that fat after digestion it will form the triglycerides. The triglycerides onwards again that is converted into the fat, storage fat, that process we call as a lipogenesis. These 4 words are very very important, need based questions, maybe chances. Here glucogenesis, first one glycogenesis, second one glucogenolysis, and then gluconeogenesis, lipogenesis. Process you should be remember, okay. And then next one lipid metabolisms. In the lipid metabolism, here triglycerides converted into the fat, lipogenesis, that one. Lipid metabolism's time synthesis of cholesterol and triglycerides. Triglycerides are metabolically active, cholesterol is reserved fat. Okay, when our body not yet have sufficient food nutrition, on the time these cholesterol used for the energy wise. Because of here already I said gluconeogenesis process time other than glucose particle in our body, fat is there, we are not yet taking any food, that food is onwards, that fat onwards, reserve fat onwards, glucose will produce. That process is we called as a gluconeogenesis. Then lipid metabolisms, okay, conversion of the lipid products, okay, producing. And then very most important function, one thing, deamination. Amino acid theriyum kada nana. In that amino acid it contains the amine group name NH2, correct na? Removing of the NH2 from the proteins. Protein is nothing but a sequence of number of amino acids are binding with the peptide bonds, correct na? So that amino acid onwards amine group removing, in the form of the ammonia. How ammonia will form? Here NH2 is there, pointer place paranganana. This is amino acid group, okay. Acid group means COOH, amine group means NH2, that both are containing, so that's why we call it these one amino acid. Here amine group NH2, acid group COOH. From the COOH onwards, this H will combine with NH2, NH3 will form, that only ammonia, okay. Now that ammonia removing from the these amino acid onwards, that process we call as a deamination, okay. Not only that, after forming of the ammonia is a very toxic substance, very toxic. It should not preserve in our blood. If it is present in our blood means our blood will be lose their efficiency of the binding efficiency. So that's why that ammonia converting into the urea, our nitrogenous waste material. Our nitrogenous waste material, what is the name? Urea kada. We are all excreting the urea. That urea is formed from the ammonia with the help of a process. That process we call as a ornithine cycle. That ornithine cycle also occurs in the liver, okay. That is one of the function. And then one more important thing, lactic acid converted into the glucose. In 10th standard you studied anaerobic respiration. Anaerobic respiration time, when the oxygen is not yet sufficient in our body, and the time when we are doing the more workout with our muscles, in the muscle cells lactic acid will accumulate, correct na? That lactic acid causes the severe pain, okay. That lactic acid want to convert it into the glucose. Lack of oxygen, that will pyruvic acid is converted into the lactic acid. So here these lactic acid converted into the glucose in the liver, that process we call the Cori cycle. Nana, here one more time I am repeating. When our body with muscles, when we are doing the over exercise, in that muscle cells, lack of sufficient oxygen, glucose is converted into the lactic acid. That lactic acid want to convert into the glucose. That process occurs in the liver. That process we call as a Cori cycle. So here these Cori cycle also one of the event in the gluconeogenesis, because of lactic acid belongs to acid group. Then onwards it producing the glucose. So other than carbohydrates onwards, any glucose substances are producing, that we called as a gluconeogenesis. That is a lactic acid glucose, conversion of the lactic acid into the glucose with the help of a Cori cycle in the liver. That is one of the function. And then next one, detoxification of the toxic substances. Detoxification means removing of the toxic nature substances from our body. Okay, for example, in our food any toxic substances are mixed unnecessarily, unwantedly. Okay, unknowingly that toxic food containing food item we ate, of that toxic food substances are entered to our stomach places, and then it will reach the liver, at the place of the intestinal places. So that digestion after, what we are taking food, that digestion after with the help of the hepatoportal system. Mammals general characteristics time I said hepatoportal system in between of the intestinal to liver, correct na? So digested food totally first it will enter to the inside of the liver. That liver act as a checkpoint. In our food any toxic substances are there, that toxicity will be removed by these liver. In this time I will tell one of the important thing in general activity of the human life. Who are more adapted to alcohol abuse, on that persons, severe persons, majority of the persons are suffered with liver hepatic problems. Liver will damage because of alcohol is a toxic substance. When the persons are taking the overdoses, that toxic substances are accumulated, are affected, show that they are affected on the liver. So oftenly that liver will be removing the toxic substances. Okay, by that chances it causes the something liver cancer or liver part will damage. Okay, so that is a basic thing in the alcoholic abusing persons. Okay, detoxification of the toxic substances, this is a function controlling by the liver itself. And then it is a one of the thermoregulatory organ. Our body want to maintain the constant temperature, homeostasis, homeothermic. Our body temperature 37 degrees centigrade. That 37 degrees centigrade totally controlling by the muscles. And totally controlling by the muscles means controlling by the muscles. And sometimes any fluctuation is happen in the environmental, external environmental, muscles will be uncontrolled shaking will be there, temperature will be gained, correct now. So along with that liver also play an important role in the maintenance of our body temperature. So that's why here thermoregulatory organ we called as a liver is a thermoregulatory organ. And then next one, hemopoietic organs. Hemopoiesis means what is a blood production, correct na? Hemo means blood kada. Hemopoietic organ in which condition, when the embryonic developmental stages, fetal stage is there na. In the fetal stage blood will be formed from the liver, so that's why in the fetal stage that we call as a hemopoietic organ. But when the fetal stage onwards, when the baby delivered after, liver will not show anything erythropoiesis. Okay, it is a bursting of lifespan completed RBC will come to the in the liver, so that's why erythroclastic organ. In the adult one it is act as a erythroclastic organ, extract the hemoglobin from the RBC cells, correct na? Yeah, here already I said bilirubin, biliverdin are formed from the breakdown of the hemoglobin. So that is one of the function in the liver, okay, by the liver. And one more thing, liver cells synthesis some of the plasma proteins, clotting factors, blood clotting factors and anticoagulants. See now, plasma proteins, what are the plasma proteins are synthesis by the liver? Albumin and globulin. Albumin and in the blood concept explanation time I said plasma proteins irukku in our blood, that plasma proteins are albumin and globulin. Globulin play an important role in the antibodies production. Albumin osmotic concentration. Osmotic concentration it maintains the for purpose of the maintenance of hemostasis. And then clotting factors, when our skin or our organs of our body any damaged, from the damaged part onwards blood will bleed. Continuous blood is inhibited by the process of the blood clotting process. That clotting process need some of the clotting factors. What are the clotting factors here synthesized by the liver? Fibrinogen and prothrombin. Fibrinogen and prothrombin. These are the two important protein substances, these involving in the clotting, blood clotting process. So that two substances are synthesized by the liver. Along with that anticoagulant. Anticoagulant means it stops the blood clotting. Because of when the blood flow in the inside of the blood vessels, blood should not coagulate. Because of our blood containing the heparin substance. These heparin substance to prevent the blood clotting. When the blood is exposing to the external environment, automatically it will start to the coagulation process. Okay, so in generally our blood containing the heparin. These heparin act as a anticoagulant. Okay, and one more thing, in the a special cells are present in the liver. That cells are Kupffer cells. Kupffer cells. These Kupffer cells play an important role in the killing of the bacteria. So that's why we call these cells as a hepatophagocytic cells. Hepatophagocytic cells. Because of these cells belongs to liver kada. Liver also another name hepatic. So Kupffer cells play an important role in the phagocytic cells. Hepatic macrophages. What is the significance of the macrophages? You know, okay, macrophages are generally present in our blood, play an important role in the immunity to engulf the dust particles or bacterial contamination. Okay, so up to now we completed the several functions of the liver. Firstly, I will be recalling. See here function first one, bile synthesized by the liver. That bile contains the bile pigments, bile salts, bile pigments, bilirubin and biliverdin. These are producing by the breakdown of the hemoglobin. Bile salts, glucocholates, taurocholate. That glucocholates, taurocholate may be sodium or potassium. Okay, and then it is a play an important role. Liver play an important role. Carbohydrate metabolism, carbohydrates products and their derivatives it will produce. One of the event in the metabolisms of the carbohydrates, glycogenesis. Glycogenesis means glucose onwards glycogen formation, and then glycogenolysis, glycogen breakdown after it will produce the glucose, and then gluconeogenesis other than carbohydrates particle, and that means amino acids and fats onwards glucose production, and lipogenesis. Lipogenesis means after digestion of the fat, triglycerides will form. That triglycerides again converted into the fat in the reserved food item. Okay, reserved storage. Okay, and then lipid metabolism also done by the liver. That time synthesis of the cholesterol and then triglycerides. Here already I said here triglycerides converted into the fat. That is one of the lipid metabolism. And then deamination, removing of the amine group from the amino acid, that we call the deamination in the form of the ammonia. That ammonia is a toxic substance that it will convert into the urea with the help of the ornithine cycle. This what is very important, ornithine cycle. And then lactic acid in the muscle producing lactic acid converted into the glucose in the liver itself. That process we call as a Cori cycle. Okay, ornithine cycle we should be remember and Cori cycle we should be remember. And then liver play an important role in the detoxification organ. It is removing the toxic substances from our food items, and then it maintain the body temperature. So that's why here thermoregulatory organ. And then it when the baby in the growing in the fetal stage on the time, these liver play an important role in the production of the RBC cells. So that's why blood. So that's why hemopoietic organ in the fetal, but in the adult stage it will not produce the blood. So that's why we call it as a erythroclastic organ, destroying the RBC cells. Okay, and then next one, synthesis of several proteins. Some of the play an important role in the immunity system, that is a plasma protein, some of the blood clotting, some of the anticoagulating. Plasma proteins are albumin and glucose, and clotting factors are fibrinogen and prothrombin. Anticoagulant is a heparin. Along with hepatocytes, Kupffer cells or special cells are present in the liver. Those things are play an important role in the phagocytic nature. So that's why these cells we call the hepatic macrophages. Okay, then up to now liver cells functions completed. Then another accessory glands, that is a pancreas. These pancreatic glands second largest gland in our body. Okay, first largest gland is the liver, second largest gland is a pancreatic. It is a mixed gland. Why we call it a mixed gland means pancreatic, some of the part act as a exocrinal, some of the part act as a endocrinal. Okay, it is present in the place of the duodenum place. Okay, duodenum shape is a U shaped. Okay, and that please recall the previous diagrammatic explanation of the digestive system on the time pyloric stomach onwards small intestinal will produce origin, that small intestinal first part duodenum, that duodenum U shape, from the U shape that place leaf like pancreatic organ we seen in the diagrammatic explanation. So now come to the pancreatic gland contains the two parts, one is the exocrinal part, another one is the endocrinal part. Exocrinal part particularly we call as a pancreatic acini, and then endocrinal part we call as a islets of Langerhans. These pancreatic acini onwards a juice will synthesis, that juice we call the pancreatic juice. That pancreatic juice pH 8.4. That pancreatic juice contains the sodium bicarbonate. Sodium bicarbonate is a basic condition, so that's why here pancreatic juice maintains the base pH, alkaline pH 8.4, above the 7 alkaline only. So sodium bicarbonate it contains trypsinogen. Along with trypsinogen, chymotrypsinogen is also is there. Trypsinogen and chymotrypsinogen inactive in form. After activation, these trypsinogen onwards trypsin will form, chymotrypsinogen onwards chymotrypsin will form. Now along with these pancreatic juice contains the carboxypeptidases, then pancreatic lipase and pancreatic amylase. Okay, one of the amylase we seen in the salivary, that we call as alpha amylase or ptyalin. Here pancreatic amylase. Okay, pancreatic lipase, then nucleases. Okay, digestion of the nucleic acids. So these substances we call as a nucleases. These nucleases are DNAs and RNAs. These are with the presence of these nucleases, nucleic acids will digest, and then pancreatic lipase, lipids will digest. That lipid digestion lipase we call as a lipase. Your pronunciation is a lipase. Stepsin, also called as a stepsin, another name. So these are the exocrinal part of the pancreatic, and endocrinal part of the pancreatic we call as the islets of Langerhans. That islets of the Langerhans, alpha cells and beta cells. The alpha cells, beta cells. Okay, so here alpha cells, beta cells onwards, alpha cells are producing the glucagon and beta cells are producing the insulin. Okay, up to that enough. Endocrinal part we will discuss in the continuation videos in the one of the chapter chemical and coordination time we will discuss. Okay, so what we completed, digestive glands we completed, salivary glands onwards, saliva, what it is contains, composition of the saliva we see. Saliva pH 6.8 and gastric glands onwards, gastric juice will produce in their 0.8 to 1.9 pH, and then intestinal juices onwards, intestinal glands onwards, intestinal juice that is contains also called as a succus entericus. The pH is alkaline nature somewhat, and then liver functions we see, and then pancreatic converts, pancreatic juice. Okay, in the continuation video I will start about the physiology of digestion. The physiology of digestion mean what we are taking food, what we are taking food, that food items converted into the simple organism, complex substances converted into the simple substances. Nothing but in the process of the physiology wise, this type of definition, non-diffusible food, that means that food is not directly enter into the blood. For absorbing process into the blood, that food items want to convert it into their simple in form. Okay, so non-diffusible foods is converted into the simple diffusion substances that accomplished by the mechanical and chemical digestion. mechanical digestion in the place of the mouth buccal cavity, sorry, in the place of the buccal cavity and the place chewing of the food, that is one of the mechanical work, and churning of the food after swallowing food, that food will be going to the esophagus, that food will be going to the esophagus with the help of the peristaltic movement. So that peristaltic movement create the churning movement, or stomach also showing the churning movement. Okay, so that play an important role in the mechanical digestion. And chemical digestion wise, several enzymes. What are that enzymes? What we studied here, digestive glands onwards, digestive gland juices are producing now. That contains several enzymes. That enzymes reactions on our non-diffusible food items, I mean complex substances, with the help of these enzymes are converted into the simple diffusion by the process physiology of digestion. In the next video, I will start physiology of digestion. Okay.